Yeah, plants respond to their environment. And a little later, I'm going to show you a video called The Intelligence of Plants. And what makes you and I intelligent? What allows us to respond to our environment? We have two systems of communication. What are those systems of communication? I'll just put the chat over here so I can see. What is an animal's system of communication within its own body?
Nervous system?
Yes, the nervous system is one system. It's an electrical system. Would you consider that plants have an electrical system? We'll investigate that today. And what is the other system that animals use to communicate? There's another system. Your brain releases these to your bloodstream.
Is that the brain
Chemicals.
releases it into the bloodstream?
Yes, and other parts of the body too, actually. There's about 8 or 9 different glands that release hormones. Yeah, hormones into the bloodstream, and that's another form of communication. Well, and it turns out plants have those too. They have hormones. But they're rooted in one spot. So there's a big difference between plants and animals. I mean, some animals are sessile, but at least their larvae can move around. And for plants, of course, their seeds can move around by wind or other methods. But for the most part, they're just rooted in one spot. They don't have any eyes or ears or a nose and no brain. So plants, nevertheless, have to sense their environment to grow and prosper. So it is important for plants to see light, of course. Light. Gravity, sure. Wind, is wind important to plants? Yes. Pathogens? Yes. Plants sense and respond to their environment. And they respond by some kind of action or movement. They can change, for example, their growth pattern. Plants have something very interesting that animals do not have, and that's called indeterminate growth. They can grow throughout their lifetime. And what is it that plants see? They might not see a predator coming towards them, but they may be able to sense a predator coming towards them, maybe through vibrations in the soil, although I've never heard of that. But they can definitely sense when a herbivore starts to nibble on their leaves, and then they produce more toxin, so they can respond to touch. How do they sense light? Well, of course, plants are interested in sunlight. Sunlight, the kinds of light that are going to help the plant photosynthesize. Plants are autotrophs. They need to make their own sugar. Sugar is not something that they can absorb from the soil. They can absorb lots of other things, but not sugar. So let's look at sunlight. Sunlight is different wavelengths of light. When combined, it looks white. But when refracted by different amounts, we see different colors. And those are the different wavelengths of visible light. So here's a rainbow, and the rainbow acts like a prism because of the droplets of rain. So there's lots of wavelengths of light. This is a spectrum of visible light. It's between ultraviolet on the smaller wavelength side and infrared on the longer wavelength side. So infrared is another wavelength you might know of as heat that exudes from the body is infrared. Ultraviolet are rays produced by the sun, and they're such small wavelengths that they can be damaging to your skin. They can also be damaging to a plant. However, when we get to reproduction, it's very interesting because flowers have ultraviolet wavelengths that some bees and other insects can see, but we cannot. The visible light wavelengths go between 400 or so nanometers to the smallest blue wavelengths to about 750 nanometers. That is their amplitude. And those ones are larger. So like about light, well, the reason the sky is blue is because all these other wavelengths here, they just go around the particles in the sky. But blue is so small, the wavelengths, that when it hits a particle, it bounces off. Ooh, I should make that blue. Bounces off these little teeny particles because the wavelengths are so small, so the sky appears blue. Do you think blue light is important to plants? Well, let's see. Photosynthesis is conducted by enzymes, but also by the beauty of pigments. And pigments are the reason that you and I can see. The rods and cones, well, the cones in our eyes have various pigments. And the beauty of pigments is that when light hits them, they will absorb some of that light and they will refract some of that light as well. And the pigments that plants have are chlorophyll A, chlorophyll B, and carotenoids. This top peak here is the peak absorption amounts that those pigments can absorb. So you'll notice that they're absorbing, blue down here, a little bit of the green, but for the most part, don't absorb green. What happens to the green? It gets refracted back at us. So that's why plants are green and the world is essentially green. And these wavelengths are very interesting too. Infrared and far red. Those are distinguishing wavelengths that affect plants very strongly. Not in photosynthesis, but in other things like growth patterns, germination, and photoperiods. So when to flower? How does a plant know? Should it flower in the spring, summer, fall, winter? A lot of choices. So chlorophyll A and B absorb blue light, absorb red light, 680 to 700, and reflects. I should have said reflects, not refracts. Reflects green light. So plants appear green. So these are some plant beans that are growing toward light. They're going toward light. How do they sense it? How do they know the direction that light comes from? Well, we can study light perception in plants. Here is a study using a part of a plant known as a coleoptile. And that is this sheath here. That covers an embryo, a growing plant, from germination so that it gets through the soil without getting damaged. Here's the first leaf. And it won't be damaged because it's been protected by the coleoptile. You find it in monocots. Grasses, for example, corn. It's a hollow cylindrical structure and it protects that primary leaf as it pushes through the soil. And so those have been used to experiment and see how plants can sense light and grow toward it. So we can test different wavelengths of light. Moving toward light is called phototropism. It's a directed movement. Tropism is a directed movement. Phototropism is a directed movement either toward light, that's called positive phototropism, or away from light, that's called negative phototropism. And phototropism is a response to blue light. That has been found through experiments. And blue light is also used in photosynthesis. Here's the apparatus that is used for studying coleoptiles. You put the coleoptile in here, plus some light filters. You can take a camera over a period of time and see what happens. What other responses do plants have to light? Well, some plants prefer full sun. You've probably been shopping for plants in a plant store and you're like, well, my balcony faces south. I'm going to need a sun plant. Or you're going to be like me, my balcony faces north-ish, northwest. When the winter, there isn't any light coming in really. So I would prefer a shade plant. What's the difference? Plants prefer full sun, get more red light. That's the 660 to 700 nanometer wavelength of light. They thrive in that light spectrum. But some plants prefer shade. Shade has more far red light. And that's because that is the wavelength of lights that basically come through leaves. So if we're talking about where plants grow, well, where they evolved to grow in the first place anyway, that would be in wooded areas or shrubland areas where there are plants above them. So the light that they thrive in is the kind of light that comes through the leaves, and that's far red light. How do they respond to far red light, which indicates shade? Well, here's an experiment. H naught or H, that's the null hypothesis, always means there's nothing interesting going on. So in other words, sun and shade plants in this case respond the same. But our alternative HA hypothesis states that sun and shade plants will respond differently to shade or sun. So simulating deep shade, we've got a lot of far red light. We're adding a lot of far red light. They all have normal light. We're adding far red. Medium, some far red. For the open sun, we're simulating no far red. And then there are different plants under these lights. Some of the species are from a normally sunny habitat, and some species are from a normally shady habitat. So we've put sun plants and shade plants in different treatments. How will they respond? Well, how can we measure their response? What should we measure? What should we measure? So when you're doing your experiments, this is one thing you will need to ask yourself. What will I measure? And in this case, we're going to measure stem growth. That's always a good thing to measure. It's quite easy to measure. And the plants that we're using in class are fast-growing plants. So here are our results. Take a look at these. This is, let's look at our sun plants. These are the plants that normally live in bright sunlight. This is the rate of stem elongation. So how much they grow, the rate. This is deep shade here. This is open sun. These are our sun plants. Tons of growth here in the deep shade. Much faster growth. Let's take a look at our shade plants. They haven't changed much. So deep shade, maybe a little bit of less growth in open sun, but not significant. What does that tell you? Our conclusions are, so plants can sense far red light. And some plants respond by elongating their stems faster. Let's see what that looks like in actual plants. Here are beans. Beans are sun plants. Here they are. These ones here. Grown in the dark. This side is dark grown. This side is light grown. Well, the light grown ones are healthy looking, not very tall. But the dark grown ones are taller. They don't look great. But essentially, they're trying to grow out of the shade. So plants, they're reaching for the sun. And unless they're shade plants, of course, but sun plants, they will always reach for the sun. How do plants know when to flower? A plant is asking itself, when should I flower? Well, when would be a good time for a plant to flower? When do plants flower? When do plants flower? For the most part. Spring, yeah. And what's happening in spring that's conducive to reproduction? You know what they say. Birds do it, bees do it, even educated fleas do it. Let's do it, let's fall in love. And that happens in the spring. So, well, what's happening now? What's happening now that's making you happier? I know it's making me happier. And that is that the days are going longer. And there's really nothing like spring to make you feel great because the days are going longer. I don't know about you, but I love that. Like this morning, I was up at 7:20, I think, and it was already light, beautiful out. And I'm like, woohoo, love that. So yeah, it's spring, the days are longer. If the days are longer and it's warmer, plants will be able to have more energy for flowers. I mean, flowers take energy to grow. They're an extra part of the plant. Plus, they're going to be growing seeds and making seeds, and it all takes a lot of energy. So yeah, spring, great time to flower. Also, these lovely, lovely creatures are around. The wasps, the bees, a lot of other animals pollinate. Flowers, flies, beetles. Beetles are big pollinators. Well, how does it know? How does the plant know that it's spring? The days are getting longer. So it has something to do with day length. And that's known as photoperiodism. That's a response based on the relative day and night length. But the question is, do plants respond to the length of the day or the length of the night? What do they respond to? And how could we tell that? Well, let's see. So this indicates a long day, and this indicates a short day. So this plant has flowered on a long day and not on a short day. This flower has flowered on a short day. And these ones are day neutral. They don't care. Well, this is weird. So these are plants that have had some light added to their night. Just a flash of light. I'm not actually sure how long that flash lasts for, but since it's called a flash, I would say it's not very long. Maybe just a few minutes. So this is a short day, but here, short day, but a flash was added at about, let's see if that's 12 hours. That's 18 hours here. So that's at about 14 hours. So at about 14 hours. And look at that, it flowered as if it were a long day. So plants appear to sense the length of the night. How can we test that? Well, this is a test that was done with red light and far red light. And it's very interesting because these are long day plants and these are short day plants. So without any additional light, as you would expect, the long day plant flowers with long day and the short day plant flowers on a short day. So that's what you would expect. And then this flash of light that's added to it at about 14 hours, in this case, is red light. Is there any difference between what you would expect normally? Yes, because look at that. Our plant that should not have flowered, flowered. So there, it does make a difference. But look what happens when we add a flash of far red light. The opposite. Our plant that would have flowered with only the red light doesn't flower with the far red light flash. And it's kind of like red has an effect, but far red turns off that effect. So it's not just any light, it's the type of light that's applied. Yeah, one more thing, and I'll talk to you about the mechanism. How do these different types of light, remember red light is direct sunlight, far red light is sunlight that comes through leaves, so it's more muted, I guess, kind of shade sunlight. How does it affect germination? Well, lettuce seeds, which I found out when I had a little 3x6, 3x6 plot of a community garden. Very small, but it's amazing how much you can grow in such a small space. Anyway, I had to plant lettuce, and you get the little lettuce packet, you know, and it says on the back, you've got to plant it only this deep in the soil because it's got to get sun or it won't germinate. So there are seeds that require light to germinate. You can't be planting them, you know, even 4 inches into the soil. They have to be up in the top inch or something. So lettuce seeds are a good specimen to try this experiment on. So they were exposed to flashes of light containing one of 2 wavelengths, the red or far red. Those are 2 different wavelengths. And after they were exposed to light, they were moistened and held in the dark for several days to see whether or not they would germinate. It turns out that red light promotes germination and far red light inhibits germination. So here's your light exposure. And this is the percent of seeds in the samples that germinated. No light exposure, only 9. Red, 98. Red and far red, half that amount. So every time that red light was the last wavelength to be flashed at the seeds, they had the most, even sometimes 100% germination. So far red tells the plant it is in the shade. No point germinating in the shade if you're a sun plant. Now, what is the mechanism? How do plants sense light? Well, it's a molecule, as you could imagine, a molecule called phytochrome. It's not chlorophyll, it's phytochrome. It is a pigment, but it's not chlorophyll. But like pigments, it changes its shape. The changing shape of molecules is really what makes the function of proteins and enzymes and therefore life possible. It's very interesting. So there are two shapes. There's a shape that stimulates germination, and there's a shape that inhibits germination. So when there's far red light, the shape will change to the inhibiting shape. If there's red light, the shape will change to the promoting shape. Very interesting. So
sorry, quick question.
Yeah.
I'm a little confused about that part. Didn't we say that when there is a red light, then there will be an increased percentage of germination as opposed to far red light?
Yes.
Why isn't it inhibited now with the red light?
This is showing that this is not well labeled. That's not well labeled. What this means is that red light has hit the phytochrome. It's changed shape to this change, to this shape here. I'm not sure why it's labeled like that, to be perfectly honest. Yeah. So it's this shape, which is the red light is responsible for the shape that stimulates germination.
Oh,
Yeah.
no, okay. Yeah, it does make sense. I actually
Just ignore those
quite
guys.
look
Yeah.
at I didn't quite look at the arrows.
Yeah.
Oh, okay. Yeah. Okay, that makes sense.
Okay, good. Yeah. When
Yeah.
I first saw it too, that like written like that, I had the exact same question. It is a little bit confusing for sure. And then this one is the same. It's the same. Yeah. Red light comes in, changes shape. I don't know why they have these abbreviations there. Changes shape, and then germination is stimulated. So red light means go. And far red light means stop. It's kind of like an off switch. Well, how was it discovered? 1959, scientists isolated a lot of proteins so that in the same kind of way that it was found that DNA is the molecule that confers traits by genes and not proteins. So it was found that one of those isolated proteins was photoreversible, which is awesome. Plants, indeed, are creatures of light. Chlorophyll is a pigment. In chloroplast, it absorbs blue light and red light for photosynthesis. Phytochrome senses shade and exhibits photoperiodism, or it rather directs photoperiodism. It senses both red and far red light. It affects other things as well. It affects flowering, timing, seed germination, also stem elongation, perhaps even stomata opening. Phototropins, they're in the cell membranes of shoots, and they detect sunlight. They respond to blue light. And when they do so, they cause the elongation of cells so that they move toward light. And there's other pigments as well. I wanted to show you something. Sorry about this, going back to the coleoptiles here. So when, oh no, that's not a good drawing, wait. So here is a plant stem with cells on one side. And then with light, the stem starts to grow in this direction. And the signal causes some of these cells to elongate. And that's what causes the bending. And that is triggered by blue light. What happens when, sorry. Oh no, I lost it altogether now. Come back. What happens when blue light hits the cell? Here's the signal of light. Here's a receptor protein. It changes in response to that signal. Another attached protein called a kinase catalyzes phosphorylation. This is ATP giving up a phosphate group, resulting in phosphorylation, adding a phosphate to a protein, and that phosphorylated starts the response. And the response is through different pathways. One response may be the activation of DNA or the repression of DNA and transcription. One response may be changing the ion flow through a channel or pump, which will change the reaction of the cell. And one may be to activate or repress translation of messenger RNA. So there are different sequences of events after blue light hits the cell wall. All right, I'd like to stop there for now. Lecture number 1.